to answer this question, usually what you will be first looking for as information about those genes, it's where and when the gene is expressed. Okay, a plant gene may be expressed constitutively in any kind of cell during any kind of developmental stage, or it may be expressed specifically in one plant tissue at a certain time of plant development. So, of course, this information will be very, very useful to understand the function of the gene. We may want to know where does the product of the gene localize. Remember, the gene is encoding for one protein, and this protein may be specifically localized into the plant nucleus. It may be localized into the plant chloroplast. It may be localized in the cell wall. And if you have this information, it will help you to better understand its function. We may also want to know what will be the protein with which the product of the gene will interact. What will be the phenotype of the plant if you inactivate this gene? If you inactivate completely the gene, which function will lose the plant? So there are many kinds of questions to which functional genomic tools may help to answer. Okay? We will try to use biotechnology, functional genomic tools to answer like these different kind of questions to identify the biological function of a gene. There are different kind of approach when you do functional genomics. I would say there are three kinds of approach. There is forward genetics. It's the classical approach. Forward genetic is the approach that was used, for example, to identify resistance gene, to identify some specific QTL, quality trait loci, and so on. From the function, identify on the basis of a phenotypic screen, we seek to arrive at the gene which is involved. This is forward genetic. What is a phenotypic screen? It depends what you are working on, but for example, if you are like me, a phytopathologist, you may want to screen a population of plants to identify those that are resistant to a pathogen. If you identify this phenotype, it's all the visible character of an individual. In this case, it will be resistance toward a pathogen. You identify this phenotype in one specific population of plants. You will seek to arrive to identify the gene which is responsible for this phenotype. Okay? This is forward genetics. Then you have reverse genetic. And reverse genetic is the opposite. You have knowledge on a genome, but you don't know what are the functions of the different genes. And the idea here is to mutate one specific gene and to see what will be the phenotype of the mutant. Okay? So it's completely the opposite, is that you have one gene, you want to know its function, you will mutate it, and you will try to see what happens when one gene is removed or inactivated. Maybe the plant will become resistant toward the pathogen, or maybe it's the opposite, it will become susceptible to the pathogen. This kind of reverse genetic approach will help you to understand the function of a gene. And then there is a third way, is to do random mutagenesis. Okay, you will not target one specific gene, you will do mutagenesis to mutate in one plant population as many genes as possible. And you will try to identify some specific phenotype associated to this mutation. So somehow random mutagenesis will be asking to do both forward and reverse genetics. Okay. So we will be speaking about different tools of forward genetics, reverse genetics, random mutagenesis to analyze different things. And the first thing we will be speaking about is how we can analyze the gene expression profile. The analysis of the gene expression profile will allow you to determine where and when a gene is expressed, under which condition it is expressed. So it will give you an information on the spatio-temporal expression of the gene. Okay, it's to know when, where, under which condition a specific gene is expressed. So this is somehow when you identify one gene, you want to know in which condition it is expressed. So it's a tool of somehow reverse genetic. You will not mutate the gene, but you will try to determine when this gene is expressed. And to be able to answer this question, when, where, under which condition a gene is expressed, you will use the reporter gene method. In most of the cases, that's the method which will be used. So you will need to use a reporter gene. So it's a gene encoding for a protein that will be easily detectable in the plant. And this reporter gene should not be present in the plant before you will transform it. Because you will do a GMO plant to be able to study the spatio-temporal expression of a plant gene. The idea is to make a chimeric construct that will be a fusion between the promoter of the gene of interest and a reporter gene. So typically, you will generate a GMO. So you will need to engineer a tDNA construct. And this is the kind of tDNA construct you will engineer. You will have, of course, a left and a right border. In this tDNA, you will need to put a selection gene, so a gene allowing to select transgenic plants that you usually express under a constitutive promoter, so a 35S promoter. And there is something which is not apparent on my drawing here. There is, of course, a TNOS terminator. And then upstream, you will have the chimeric construct necessary to analyze the function of a gene. So you will have the promoter of the gene of interest and it will be used to control the expression of a reporter gene, and after you will have a TNOS. So you see that here you will not use a P35S promoter or an inducible promoter. You will use the promoter of the gene you study. You will clone, amplify, clone, insert into the tDNA the promoter of the gene for which you want to know when, where, under which condition it is expressed. And this promoter will drive the expression of a reporter gene. What will be the reporter gene? It may be of different kind. The most commonly used reporter gene is what we called the GUS gene. It's a gene which is encoding for an enzyme which is called the beta-glucuronidase. It's an enzyme of bacterial origin, and this enzyme encodes for a protein, which is able to act on different substrates, and it acts more particularly on X-gluc. And when it degrades X-gluc, it's making a blue precipitate. So you will generate transgenic plant expressing the GUS gene under the control of the promoter you study. And those transgenic plants, you will expose them to the substrate of the beta-glucuronidase. The beta-glucuronidase produced only in the tissue in which the promoter is active will degrade the X-gluc, forming a blue precipitate that you can observe under a microscope. And you will know in which type of cell the promoter is active. So here you have different examples of genes that are specifically expressed in one root tissue. It may be the cell, the endodermis, the cortex, the epidermis, it may be the root cap. And you see that the profile of the gene expression is different each time. And the expression of each gene is driven by a different promoter. The fusion between this promoter and the beta-glucuronidase, the GUS gene, allows us to determine in which type of cell the promoter is active. Where you have no blue precipitate, it means that the promoter is inactive. There is no expression of the reporter gene. Where you have the blue precipitate, it's where the promoter is active. So this will help you to determine in which type of cell the promoter is active. And it will give you a very, very important information on the function of the gene. The advantage of the beta-glucuronidase is that you may use different substrate, or the X-gluc or the MUG, that will allow or a qualitative analysis of the promoter function or a quantitative. In this case, you will have formation of a fluorescence that you can quantify with a spectropluorometer. But the principle is always the same, is that you will drive the expression of the beta-glucuronidase with the promoter that you are studying. Where the promoter is active, you have production of the beta-glucuronidase. When you expose to a substrate, you can detect the presence of the beta-glucuronidase. Another kind of reporter gene is the luciferase. The luciferase gene, or LUC gene, is a gene which is normally present in a firefly, Photinus spiralis. And it's degrading a substrate which is a luciferin. And when you have degradation of the luciferin, you have light emission. So you can detect under a microscope the light which is emitted where the promoter is active. So it's the same principle. It's just that the reporter gene is different. So to use luciferase, you need to have quite very expensive, very particular technical equipment. You need a specific microscope. The advantage is that you can quantify, you see, according to the light which is emitted, you can quantify the level of expression of the reporter gene. Here, when you use the GUS is not quantitative at all. You cannot quantify the intensity of the blue. You can only detect presence or absence of the blue color. Here, of course, you can quantify, but this is an extract, protein extract, so you have no idea about the cellular specificity of the activity of the promoter. A third kind of reporter gene is the GFP, the green fluorescent protein. It's a protein which is normally present in a jellyfish, Aequorea victoria, and this protein is, the GFP is fluorescent under some specific condition. If you excite the protein with a specific light, it will emit a green light that you can observe with a fluorescent microscope, a confocal microscope. So this is a very useful reporter gene, because at the opposite with the beta-glucuronidase, here you do an histochemical analysis on a tissue, but it's not a living tissue. You cannot do the observation in vivo. You will need to expose the plant tissue to a substrate of the enzyme to observe a precipitate, but the plant cell will not be alive anymore. At the opposite, here you can work in vivo with living cells, and you don't need to add a substrate. You can detect directly the presence of the fluorescent protein, which is a product of the GFP reporter gene. Okay, this is really the use of the transcriptional fusion, we call it, when you fuse the promoter of the gene of interest under the control of the promoter of a reporter gene. This allows to analyze the expression of one specific gene under certain condition. Okay, it's to do analysis of the function of one gene. Of course, there are now a lot of tools that allow to analyze the level of expression of any kind of gene under certain conditions. It's all the transcriptomic approach. And particularly since we can do high throughput sequencing, you know that we can sequence easily the complete transcriptome of an individual. And for plants, it's becoming cheaper and cheaper, and you can easily have access to the expression profile of all the gene of an individual under some certain condition. It's all the RNA-seq approach. But to use those transcriptomic approach, those high throughput sequencing approach, you need to have the information on the genome sequence of the organism, the plant you're working with. Otherwise, you cannot, because you will have sequence of transcripts, but you will not be able to align them to a reference genome. If you have available the complete genome of one plant species, you can envisage a full transcriptomic approach, and analyze in one plant under which specific condition all the plant genes are expressed. Here, this approach I described you, it's to analyze the spatio-temporal expression of one specific gene. It's based on the production of a transgenic plant carrying this construct. But these GMO plants, once you isolated a population of a GMO plant carrying this construct, you can expose those plants to any kind of conditions and see under which condition this specific promoter is active. Did you all understand here the use we can make of GMO plants to analyze the spatio-temporal expression of one gene of interest? Is it clear, this small part here? Yes? Yes? Okay. So I will continue. So we may use transgenesis and functional genomics to study the expression profile of one gene. That's what we saw. We may also use transgenesis to analyze the protein subcellular localization. So the idea is to know where inside a plant cell a protein will localize. And to do this, we will again use a reporter protein. The idea will be to make transgenic plants produce a chimeric protein that will be a fusion between your protein of interest and a fluorochrome. That may be GFP, YFP, RFP. Okay? So it may be done into stable line or in using a transient expression system, the agroinfiltration system, you remember, by infiltrating tobacco leaves with agrobacteria. Anyway, you may want to do stable lines or to work under with a transiently expressed protein. In any case, you will need to generate this tDNA construct. And I'm sorry because TNOS terminator are again missing, but you know that you will need to use the TNOS. Okay, now you start to be familiar with this kind of construct. You have a left border, a right border. Of course, a genes that will allow to select the genetically modified cells. So remember, it can be HPT, NPT2. It may be a gene coding for an enzyme degrading an herbicide, whatever. You need to have a selection gene expressed under the control of a constitutive promoter, and you will have here a TNOS. To analyze the subcellular localization of a protein, you will use the complete open reading frame of your gene of interest, and you will fuse it to a reporter gene, which is generally a fluorescent protein, the GFP. And so you will produce a protein that will be this complete protein. And this protein will localize where should go normally this protein, but it will also emit a green light if you excite it under the proper condition. Okay? Here, you see that we choose to have to use the promoter of the gene of interest, but it could also be a 35S promoter. It could also be inducible promoter. Here, the idea is not to know when this promoter is active. The idea is to know where does this protein localize in a plant cell. Okay? So the choice of the promoter is yours. It can be the promoter of the gene of interest or another one. And then you can see here some results. Typically, here you have a transgenic plant that is expressing this kind of chimera. You have production of a protein fused to the YFP, which localize specifically in the nucleus. You have a nuclear localization of the protein. So your protein of interest, you know now that it's having its functioning into the nucleus. It's a nuclear protein. Another example here, it's a fusion with the GFP. You see that the GFP localized specifically at the plasma membrane. So this protein is a membranar protein. And if you know this, it helps you to better understand the function of the protein. Here, it's a protein which associates to mitochondria, localized to mitochondria. So this kind of GMO plants help you to understand where does a protein localize into the plant cell. If you add also the promoter of the gene of interest, then you will detect the protein, the fluorescent protein, only in the cell where the promoter is active. So finally, using this construct, you will have two informations. One on the subcellular localization of your protein of interest, and one on the expression profile of this gene of interest. We'll also know at the same time when and where, under which condition this promoter is active. Okay, so you have the choice. You may only want to study the subcellular localization of the protein, then you may use a 35S promoter here, or you may want to have the two information in the same plants, and you will use to clone and the promoter and the open reading frame of your protein of interest. This is another example of the kind of study you can do. Here you analyze the localization of phytochrome B protein fused to the GFP under different conditions. When the plant is grown in the dark, the protein, the phytochrome B, is cytoplasmic. And when you expose your transgenic plant to a red light, the protein changes localization. It moves to the nucleus. Okay, so the light induces a translocation of the protein to the nucleus. And this is an information that you can get using this kind of approach. It allows you to gain amazing information on the function of the protein you study. I was speaking about the GFP here, the green fluorescent protein, but you have a lot of fluorochromes that you may use. You may use CFP, which is a kind of blue fluorescent protein. You may use a Venus, which is a yellow fluorescent protein, an orange one, a red one, a green one. Okay, and those fluorochrome will emit different light that you will be able to separate using a confocal microscope. So you can imagine transiently expressing two chimeric constructs into the same plant cell and see if two different proteins, here SMD1 protein fused to the GFP, and here the HTR protein fused to the cherry, you can see if those proteins co-localize. Do they localize to the same subcellular compartment or not? If they localize to the same subcellular compartment, it means that maybe they are involved in the same biological process. So you see, this kind of experiment gives you a lot of information. And you can imagine that you can express not only 2, but 3 different chimeric proteins into the same transgenic cell and so on. So it's a very interesting kind of approach to gain information on the function of one gene, the function of one protein encoded by this gene of interest. I want to be speaking about gene inactivation. When you study one gene, you want to know what's its function. So I would say the best way to know what is the function of a gene is to inactivate it. You inactivate the gene and you see what happens in the plant. Maybe the plant will not be anymore able to do something. So that's a very interesting kind of approach. How to inactivate one gene? I will give you the example of Arabidopsis thaliana. I told you that we could transform Arabidopsis thaliana using Agrobacterium tumefaciens using the floral dip transformation method. And you will have insertion of tDNA in some germlines in the flower buds, and you will harvest seeds from these plants that will be transgenic. And actually, in each different seed that you will collect from this plant, you may not have a tDNA, but some few seeds will give rise to a transgenic plant. The different transgenic plants will not carry a tDNA insertion in the same, at the same locus, because the insertion is random. But if the tDNA insert into the open reading frame of one gene, the tDNA is kind of big. It will completely cut the gene into parts, and it will inactivate the gene. You understand, here you have in pink the open reading frame of one gene. If a tDNA is inserted by Agrobacterium tumefaciens into the open reading frame of this gene, it will be integrated here. You will split this gene in 2 parts. It will be cut in 2 pieces, and it will not be active anymore. So this gene will be inactivated. Okay, you will tell me, yeah, but the integration is random, and we cannot say in which gene the tDNA will be inserted. What is very nice with Arabidopsis thaliana is that we know its full genome sequence. So if we sequence the part of DNA which is close to the tDNA, we will know exactly in which part of the genome the tDNA has been inserted. Okay, we will compare this sequence to the complete genome of Arabidopsis thaliana, and we will know exactly in which gene the tDNA has been inserted. It's very easy if you work with a few tDNA plant to generate a collection of thousands and thousands of mutants. And nowadays, it's very easy to tag each of these mutants and to say for each of these mutants, it's carrying a mutation in this gene, because we have all the genetic information available for Arabidopsis thaliana. You can screen your population of mutants for one specific phenotype. So it may be a developmental phenotype. You see here it's a collection of tDNA mutants. We transformed tens of plants. We harvested thousands and thousands of seeds, genetically modified, and you grow them, and you see that you have different kind of phenotype. because probably some genes have been mutated, inactivated, which are very important for one specific step of the plant development. So by screening such a mutant collection, it may be a screen for a specific developmental phenotype, but you may screen a mutant collection to identify those that are resistant to pathogen, that are resistant to salt stress, to heat stress, and so on. You can imagine all kinds of analysis, and you can identify the gene in which it has been mutated, or you can say, I want to know if there are mutants available for this gene, and ask for the mutant and study it in your laboratory. It's then a kind of one candidate approach and not a genetic screen anymore. Okay, so I told you to explain you this. Now a lot of mutant collections are available for Arabidopsis thaliana, and because we know completely the sequence of the gene, each of those mutants has been attributed a tag. We know exactly in which gene each mutant of the collection is mutated, and each Arabidopsis thaliana gene has a name. It's like this, AT1G tatata, because it's one gene which is present in the first chromosome, 1T3, and the name it's a gene which is present in the chromosome 3, and we have the exact name of the gene. So we know exactly all the genes of Arabidopsis thaliana. If I want, I ask to the people who take care of this collection, and I ask for a mutant in this specific gene. Here I want to study, and they will send me some seeds I will be able to analyze in my laboratory. Okay, that's what you see here. This is one gene, AT3G 59610. This gene, you see that there is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 different tDNA mutant lines that are available in different collections. There is the Salk collection, which is American. There is the Wisconsin collection, which is also American. The Gabi collection, which is a Japanese collection. You see that there are many kinds of mutant Arabidopsis thaliana mutant collection, and I can ask to those institutes which take care of the mutant collection to have different mutant lines that will all be mutated for this gene, which is a gene I want to study. So it's a very, very powerful tool. There is one thing I must say more. This is true for Arabidopsis thaliana. I would say that in the early 2000s, it was only true for Arabidopsis thaliana, but now we have the complete full genome sequence for many, many plants, tomato, rice, I told you. And this kind of collection also exists for other model plants, including some crops. Those collections may not be as impressive as the one we have for Arabidopsis thaliana, but still, these kind of tools exist for some other kind of plants. The limitation of this, it's gene redundancy. In plants, you may have different copy of one gene, and when you use the tDNA insertion approach, if in a gene family you have 3 different genes, one mutant will carry one insertion in one gene. This one will be inactivated, but the function will remain due to the presence of the 2 copies, if you want, of this gene. So the function will not be lost. If you want to analyze the function of this gene, which has 3 copies, you will need to inactivate the 3. And this is much more difficult. It's possible for Arabidopsis thaliana because you can cross each single mutant to make multiple KO. But you will need to do crossings, and this will require some time. So it's a limitation of the use of the tDNA mutation approach, is that a lot of genes are redundant in plant genomes. Not all of them, but a lot of them. In case the inactivation of one gene gives a strong developmental phenotype or is lethal, you will not be able to study the function of this gene. Okay, you understand. If you inactivate one gene, but this gene is instrumental to the plant survival, you will never have a tDNA mutant for this gene. And another very, very important point to consider is that often, and we don't know exactly why, but often, when you transform a plant with Agrobacterium tumefaciens, it will not carry only one tDNA insertion in its genome, but it will carry 2, 3, several. It depends. Okay? And then, if several genes are inactivated in one Arabidopsis plant, if you have 2 or 3 insertions, the phenotype you will observe, it may be due to the insertion in this gene. You will say, okay, it's this gene which is responsible for this phenotype, but maybe not. Maybe it will be due to the second insertion in another gene or in the third one. Okay, so the fact that you may have different insertion in one transgenic plant renders the functional analysis sometimes a bit tricky. So when you isolate one tDNA mutant line, you will always need to verify how many tDNA molecules are present into the genomic DNA of the plant. Okay, so we said that a tDNA may insert anywhere in the plant genome. The insertion is completely random. Sometimes the insertion will be between 2 genes. Okay, so it will not affect the function of any kind of gene. Sometimes the insertion occurs, for example, here in the middle of one gene. This very often it leads to a loss of function. Okay, the function of the gene is lost. That's what we call a KO mutant. The gene is completely inactivated. Sometimes you may have insertion of the tDNA in the promoter of one gene. This will reduce the expression of the gene. So you will have not a KO, but a knockdown. If in the tDNA you have a strong constitutive promoter, and the insertion is in the promoter, then your gene here will be under the control of this 35S promoter. And you may have an increase in gene expression. You will have a knock-on. And remember, you can have multiple KO in one plant. You may choose to generate multiple KO by crossing single mutants, and it's very often the case. So using tDNA insertion approach, you may have different finality. Okay, you may have a KO, a knockdown. You may use a specific tDNA to generate some knock-on. And do not forget that you, and it's particularly true for Arabidopsis thaliana, it's very easy to cross single mutant to generate multiple KO. So this may be very interesting to study in combination the function of different genes. Okay, this part on tDNA insertion is very important part of the lesson. Did you all understand the principle of this tDNA mutation, what we call insertional mutation. Is it okay? Or do you have, is someone having a question on this? It's very important that it's clear. Everybody understood this?
Yeah, it's clear.
It's clear?
Yeah.
Okay. So then I will continue a little. I will say only one word on gene downregulation. We may want, we so inactivate completely one gene, or we may wish to reduce the level of expression of one gene. We saw that this may be achieved by tDNA insertion, but this is kind of, you need to be lucky to identify this kind of tDNA insertion. Huh? You really need to be lucky because you will not be able to target the tDNA insertion. So you may be searching in the collection database for a mutant which is carrying the tDNA in the promoter. That's what you can see here, and check if those two lines are resulting in a lower expression of the gene here. But it may not always be the case. Huh? It's not because you have insertion of a tDNA here that you will have a reduced expression. Sometimes you have a knockout, even if the tDNA is here, okay? And sometimes you have no change at all. So it depends on the gene. So how can we do to downregulate one gene? And there is one approach which is commonly used. It's the RNAi technology approach. So the idea is to silence a gene. I will be speaking about RNAi technology during the last course, so I will go through it very fast. The idea is to make the transgenic plant produce double-strand RNA. Okay, you will integrate the ORF of the gene of interest in sense and antisense, and those will have complementary sequence, so it will form double-strand RNA. And double-strand RNA in a plant cell is recognized as something abnormal, aberrant. So the plant is having a kind of defense mechanism, and it will degrade this double-strand RNA. It will make it degraded. And it will lead to inactivation of this specific gene expression, because here you will be targeting one plant endogene. Okay? So this is the basic of RNAi technology, and this I will be speaking about during the next course. So I go through this. I want to speak about gene overexpression. So we saw it was possible to analyze the profile of expression of one gene, the subcellular localization of one gene. We saw that we could use tDNA insertion to inactivate some gene. Now we will see what can be the interest of overexpressing one gene. Okay? It's another way to study the function of a gene. A gene may be not so strongly expressed in a plant, and you may want to see what happens in the plant if you produce more of the protein encoded by the gene of interest. So here the idea is to make the plant, the transgenic plant, produce more of this protein. And for this, you will need to engineer a tDNA construct with a left border, right border, and you will express your gene of interest under the control of a promoter. again, it may be a strong constitutive promoter, an inducible promoter, or a tissue-specific, developmental stage-specific promoter. Okay, depending on what you want to do. Here, the idea is to make the transgenic plant produce more of your gene, of your protein of interest. And here is an example of a transgenic plant that you can see here that overexpress a NCED gene. And this gene is involved in abscisic acid biosynthesis. Those are wild-type plants, so they are non-GMO Arabidopsis thaliana plants. And you see that if you expose those plants to drought, the wild-type plant will die. At the opposite, a plant which is expressing NCD at a higher level will produce more abscisic acid, and abscisic acid is a plant hormone which help to resist to water stress, and the plant survive better. Okay, so you know that this gene is involved in drought stress resistant, and you may, because you know that abscisic acid is involved in the resistance to drought stress, you may suppose that the NCED gene is involved in ABA physiology. So you see that the kind of experiments you may perform using this overexpression approach. I will say something more here. We are speaking here about functional analysis of plant gene. And I told you, if you want to know the function of one plant gene, you can make the plant produce more of the protein encoded by the gene. But it's the same approach that will be used if you want to overexpress in a plant a gene which is not of a plant origin. Okay, you may want to express in plant genes that are from bacteria, insects, and so on. Remember when we spoke about the plants, transgenic plants, resistant to herbicides. And you remember there was one bacterial gene involved in the biosynthesis of amino acids that was introduced in the GMO plant for them to resist the herbicide. It was exactly the same approach. The idea is to overexpress one gene into a transgenic plant. Here, when you do functional analysis, it's a plant gene that you will make the plant produce in a higher quantity. But it can, it may be the same, I would say, approach if you want to express any kind of gene.
Can I ask about the example a little bit?
Yes.
So, as I understand it, in this example, they are overexpressing the NCED gene, and they observed that the transgenic plant survive better under water stress, right?
Yes.
So, I don't really understand whether they know that the gene is involved in ABA biosynthesis before or after the gene overexpression.
Yeah, no. In this case, well, it really depends. You are true. In this case, they knew before. And because they knew it was involved in ABA synthesis, they said, okay, most probably if we make the plant produce more abscisic acid, it will be more resistant to drought stress. Okay, and they verify it. But you can imagine the same. Huh? They may not have known, and after, by doing more investigation, conclude that it was due to this. But yeah, you're right. The way I presented it's as an example. But here the function was known before, that it was involved in ABA biosynthesis, but they wanted to show that if you make the plant produce more abscisic acid, it will be more resistant to drought stress.
Okay, I understand.
But
Because
it's,
I wasn't sure whether it was like they know the gene is involved in the biosynthesis before, or like they see that the gene is involved in water stress response, and then they conclude that it is involved in the ABA biosynthesis.
Yeah, it was the way around.
Okay, okay.
Yes.
Thank you.
But the idea was to illustrate and what kind of result you can achieve by over-expressing one gene in a transgenic plant. You see that you can gain very important information on the function of one gene. Here, I may not have been speaking about this. It was to give you one more information. But I may have concluded, if I had removed this sentence, this small thing here, if I was not having shown this, I could have concluded from this experiment that the NCED gene was involved in plant tolerance to drought stress.
Yes, I agree with that.
Yes, and that was the idea I wanted you to have in mind. That by doing this kind of approach, you can get this kind of information of one gene. So here, if we didn't know anything about it anyway, we may have concluded from this experiment that the NCED gene is involved in tolerance to drought stress. And a lot of information on plant genes have been gained through this kind of approach. You may not wish to inactivate one gene. Sometimes you will have more information if you overexpress one gene. It really depends on what is the function of the gene you study. Protein detection and purification. I will go quite fast through this part. The idea is to tell you that you see that you can overexpress one gene. It's exactly the same, it's exactly the same construct as here. But here, if I overexpress this gene in those plants, I cannot detect the protein so easily. To know if this gene is expressed in the transgenic plant, I will need to quantify the transcript. So I will need to do a northern blot, or to do RT-qPCR to quantify the transcript of the gene to know that it's really overexpressed in those plants. If I add a tag to the protein that I will overexpress in the transgenic plant, maybe I will be able to detect it more easily. Okay? By using what I call a tag, I will be able to detect the expression of the transgene. This is used to detect, quantify, localize a transgene. We have been speaking about the GFP. The GFP is the same. It's a tag. I can detect the expression of the transgene in the transgenic plant because you will have green fluorescence. But the GFP is a big tag. So sometimes it may interfere with the function of the gene, and it's interesting to do protein localization, a little bit quantification, detection, of course, but its size may interfere with the function of the gene. Same with the GST, it's a big protein, but we can easily use it to detect protein because we have antibodies against the glutathione. And then you have a lot of small tags, very small tags, 6-histidine, the flag, the MYC, the HA tag. And antibodies are available to detect those proteins, those peptides. So you will be able to detect, doing a Western blot, your recombinant protein. Also, a lot of tools are available to purify, to trap the tag. So you will be able to purify your protein of interest. So this is kind of interesting. And you can imagine that if you can catch your protein of interest because of the presence of a tag, if you work in some proper condition, you can co-purify with your protein of interest all the proteins with which it is forming a complex. That will allow you to identify possible partners of your protein of interest in one specific plant function. That's what happens when you do pull down. So for example, imagine the GST pull down. You fuse your protein of interest, that is called the bait, with the GST. And you have a system which allows affinity purification of the GST. So it will catch the GST. And together with the GST, it will catch your protein of interest and all the proteins that possibly interact with your protein of interest. And this, using a specific column for affinity purification, you will be able to isolate and later on to identify. Okay, so this allows to identify protein complex. You will be able, like this, to purify all the proteins that are interacting physically with your protein of interest. You understand this part? A tag will allow you to detect your protein. For example, by doing Western blot, it may also help you to purify it for doing something. And it may allow you to trap the protein it's interacting with. So this is also fitting with this last part of my lecture related to interactomic. Okay, this is exactly the same here. Huh? You have one, for example, tag protein, a protein of interest with a small tag. You have antibodies or beads that allow you to trap the tag, and you may catch all the partner proteins, analyze them, for example, doing mass spectroscopy, and you will identify them. If your protein is not interacting with your protein of interest, is not interacting with a protein or proteins, let's say, but more with DNA, you can also trap the DNA to which it's interacting by doing ChIP-seq. So it's another example of functional genomic tools that you may use. For example, this would be very useful to analyze the function of a transcription factor. If your gene of interest is a transcription factor, you want to know which gene are regulated by this transcription factor, you do ChIP-seq, and you will trap the promoter targeted by the transcription factor. This I will skip, and I will stop here, the lecture on functional analysis. I want you to understand well that plant transformation, genetic engineering and functional genomic tools are very, very interesting tools to study plant gene function in vivo. That's what we saw today. We can use the properties of Agrobacterium tumefaciens to analyze the function of plant genes. And you saw we can analyze the expression profile of one gene. Okay, we can analyze, determine when, where, in which condition a specific gene is expressed. We can localize the protein of interest into a plant cell. We can see what results from the inactivation of these plant genes. So genetic engineering, it's a powerful tool to study the function of plant genes. I will stop here. I want that we start the next lesson by doing a small exercise together. I will send you today a document, which will be this document with some other figures. And I really want you to try, there will be some questions. I really want you to try to work on this exercise and to answer the question for the next time we will see each other. So you will have a lot of time because the next course is the 1st of December. So you will have 2 weeks. So I will send you today the lecture we made today. I will send you today this small exercise, and please try to work on this exercise for the next time. It will help you a lot to understand all what we have been speaking about today. Okay? Yes?